Like any organ in the body, the brain is composed of specialized cells. In the brain, these are called neurons. Computer reconstruction of these cells shows that they have long extensions called dendrites that branch at various points. The cortex is divided into layers that each contain different types and shapes of neurons. While each cell is unique, they can be grouped into similar families, which are often named after the shapes of the cells. For example, chandelier cells or pyramidal cells. A census of neurons helps us predict what the composition of cell types in each layer should be. To build a model of the brain, we first create a virtual volume, in this case a column of tissue, which we then fill with three-dimensional reconstructions of neurons. This shows you how we assemble a region of the cortex that is 2 millimeters high and 0.5 millimeter wide, or about the size of a freckle. Neurons don't exist in isolation. The brain environment is more like a dense forest filled with trees whose branches are touching. There are thousands of points on each neuron where another cell can make contact to pass on a signal. Here we are showing only 10% of the neurons, because if we showed all of them, we wouldn't see any detail, just an opaque wall of cells. A cell can only form a limited number of connections, and it takes a lot of energy to produce the structures that connect cells, called spines. After we establish all the connections in the model, we can isolate a single neuron and inspect the locations where it receives input from other cells. Excitatory connections are shown in red, and inhibitory connections are shown in blue. Neurons are filled with ion channels that open and close to create the electrical behavior of the cell. When a great number of the ion channels open at the same time, we observe a firing of the cell, which results in the electrical signal or message being passed on to all connected cells. With many connected neurons in our model, we can start to see how groups of neurons contribute to network activity. Modeling this kind of complex activity helps us understand how neurons working together produce actions, memory, and higher brain functions. With the three-dimensional model, we can start to replicate experiments that were originally performed on biological tissue. The results of these virtual experiments let us evaluate how similar a model is to the real thing. Going beyond this small volume, we can apply the modeling process on a larger scale for different brain regions. We hope that with this approach we will be able to build a model of the entire brain to see which phenomena we can explain with existing data and where we still have gaps in our knowledge.